Thank you, Noah. That was wonderful. All right, the next one we have for today is Richard Masse. He's going to be doing a comparative analysis on a couple different species of surgeonfish around Hawai'i Island.
Good morning. Morning. My name is Richard Masse. I am a fisheries research specialist in the Hawaii Comparative Fisheries Research Unit. I'm also a TCBES grad. I was in the 2017 cohort. So I'm here to talk about one of our current projects, comparative life history and reproductive biology of 5 nearshore surgeonfish species around Hawai'i Island. Okay, part of the DAR's Olimua Marine Initiative is the Sustainable Herbivore Management Plan. What we wanted to do is look at life history, which is evaluating the age and growth, reproductive biology of 5 herbivore species. First one is very popular in the unit, the Acanthurus achilles, or the pakuakui. And then we also worked with Acanthurus triostegus, which is the manini, Tinacatus strigosus, which is Kole, Nasolatratus, which is the umamale, and the Acanthurus nigrofuscus, which is the mi'i'i. The first 4 are part of the herbivore Sustainable Herbivore Management Plan, and they're exploited species, but we also wanted to look at an unexploited species kind of as a control, which is the mi'i'i. Okay, our original study, which Annie talked about, was a study that we did about a year ago, which was the life history of the kukui. First thing we do is we want to look at otoliths to get age and growth. The otoliths is a little, very small acellular structure that's under the brain of the fish. We harvest that, mount them, polish them, and read them so you can see how old they are. Next thing we do is we pull out the gonads, get the histological samples processed for us, and then we get a chance to read them, look at them to see, to determine the reproductive status of both males and females. And this, at the end of the project, would give us seasonality. So, along with the pakukui, we did 2 other Acanthurus species, the mi'i'i and the manini, Tinacatus species, the Kole, and the umamale. Now these are all herbivores, they're all similarly bodied fish, so we wanted to see how they're similar and how they're different. So our methods, well, we got 20 of each species per month for a year. And we started this in September of 23, so we're about halfway through the project. So first we record the lengths and weights of each fish, and then we harvest the otoliths to get the age and growth, and then for age and length frequencies, and then calculate growth curves. And then with the gonads, we get the histology so we can do the reproductive strategies and the seasonality. So our results so far is the mi'i'i are the smallest of them. They're about 141 millimeter fork length for the males, 139 for the females, so they're pretty similar. The manini are 175 millimeters fork length for the males and 174 millimeters fork length for the females. So they're a schooling fish. They all look the same. It's better to look like everybody else and not stand out, or you get predated. The Kole are the next largest. They're 183 centimeters for fork length for the male and 170 for the female, but our female sample size is pretty small. Hopefully with the rest of the samples we'll be able to see if that trend continues. The next size up is the Kui. They're 183 millimeters fork length for the males and 191 for the females. So the females in this case are larger. And the umamale, which are the largest ones, are 291 millimeters for the males, 261 millimeters for the females. So they are, the females are actually smaller, in this case the males are larger, but they're the only sexually dimorphic species in the group where the males will be larger, a little bit more colorful, and have tassels on their fins. So looking at age, there is some similarities and differences with these as well. The Ma'i'i, the Kole, and the Menini, we've aged out for about 12, to be about 12 years old. Now this matches a lot of the publications. There's a new publication on the Ma'i'i, I think over in Okinawa, where they can be as old as 25 years. So we want to see if we can see older samples here. The Umamale, we've aged out to about 15 years. Now, there's a project over that was done in Pohnpei and Guam that aged them out to about 14 years. So that's pretty similar. But also there was one in American Samoa where they aged out to be 25 years. And then the Great Barrier Reef, they're 39 years. So we want to see if those, we can find those age groups here. Our population here, we have a little dip around 9 or 10 years. We don't know if that was a cohort or a sampling. These our spearfisher is trained to get random samples, but in this case, there's there's more females in a group than male. So we do have a few less males, but we're trying to figure out where we can go with that. The last is the Acanthoris achilles, the Pukukui, and we've aged them out to 39 years. So just looking at these 5 acanthorid species, they have a wide range in longevity. And this is something we want to see. But the other thing is reproduction. So just for context, I want to look at females. You can see the whitish structures in the posterior part of the visceral cavity or the ovaries. You can see they're fairly similar. Don't have pictures of the Pakukui, but they're very similar. So you can see they're all pretty much the same for all 5 species. However, what's different are the testes. The testes in the Ma'i'i, the Kole, and the Menini are rather large. So the white structures you can see towards the back of the visceral cavity, but they take up a much larger proportion of the visceral cavity. Now the Umamale, which is like the Pukui, we don't have a picture of, they're very, very small. You can hardly even see them at the bottom of the visceral cavity. And this is what's really interesting. And this is what I want to focus on a little bit. Now what's so important about the size of testes in the fish? Well, first thing we do is when we harvest, we harvest the gonads, we weigh them to the nearest thousandths of a gram. And we want to do that to generate the gonadosomata index, or GSI. The GSI is the gonadal weight divided by the body weight multiplied by 100 to get an index. So why is this going to be important? What's the difference it's going to do? Well, the 2 main mating strategies are group spawning and paired spawning. And in group spawning, there's no mate selection. They're just, everybody's going out, they all look the same, and they're trying to get as much their gametes out as possible. Well, if, in that situation, like the Menini and the Ma'i'i, you have to put a lot of energy into just producing sperm, because you don't look any different. So you have very large testes. That takes a lot of energy. On the other hand, if you're a parent spawner, you have to attract a mate. And you do that by having, being bigger, having more coloration, bigger fins, or being more aggressive. So all the energy goes towards that, and less energy goes to the testes, therefore, there are smaller testes. But if you're only spawning with 1 female, it's an efficient way to do it. So we're using the GSI to see if we can determine the mating the reproductive strategies of the fish. So to put some numbers to it, what we found out so far, the Pakuakui, they have a very low GSI, and that's indicative of small testes and a paired spawning. So that's suggestive of paired spawning, and you actually see this with the divers, and somewhat in the literature, it'll be you see a group of fish with a couple large individuals. Those are the spawning pairs. And same thing with the Umamale. They, so far, are trending towards very small, low GSI and very small testes. And that would be indicative of, again, paired spawning or colonial spawning. And this, you'll see, if you're going in a group, there'll be a large number of females and a male. Now, on the other hand, the very high GSI is indicative of group spawning. Okay, they need to put out as much the sperm as they can. And the Ma'i'i and the Menini, you can see have very, very large, very high GSIs and large testes. Now this is is common to the literature, and if you go out and you'll see a group of Manini and Ma'i'i. Now the colae, so far, we have very small, there's a low GSI. However, if you've seen them, they are in large groups, and you don't see very large individuals that stand out. But we're hoping getting more samples is going to help us with that. So when the whole study is done, we'll be able to look at seasonality. And the paper that it's in review now, we found that basically the Pakui, they spawn year-round. The lines represent sampling events and the proportion that are actively spawning or spawning capable. And you can see now on 75 to 100% on every single sample throughout the year and throughout the lunar cycle responding. So it's very interesting where the Pakui, they're spawning every day. Now, putting this all together, the information that we have so far is we can see differences in the 5 acantharid species. Okay? They have different longevity. We're going to see is that throughout the species or in different regions. We can definitely see that they have different high and low GSIs, which could suggest the mating system. And as we get through the project, we'll look at seasonality. And that seasonality, that information can all go to what we're trying to achieve is to provide information for the sustainable herbivore management plan that the DAR is using. And that is the goal of our project. We're halfway through it, so stay tuned. So I'll say thank you to DAR for supporting the unit, and Tim and Lillian for the support, and everybody that's helped out with fish processing, and UH for providing us with the space. With that, I'm done. If you have any questions. Okay, no questions? All right. Thank you very much.